DNA replication, when, how, and why. So, we will study today DNA replication. Welcome everyone to BioBoost channel. Today we will cover one of the fundamental processes that is included in our CSIR NET syllabus. So, what is DNA replication? We know that to continue life, cells divide. From a single cell, multiple cells get divided. This whole process takes place during cell cycle, in which we have read in our last videos that G1, S and G2 phases are there. During S phase, DNA is getting replicated. We know cells get ultimately divided into two and multiply further to bring new generations. So DNA replication is a reaction in which daughter DNA molecules are synthesized using the parental DNA molecules. Why DNA? Because it is the foundation of genetic inheritance and it carries the genes. So it also gets replicated further. In the given diagram, you can see from one parental strand, two daughter strands are made. So that's how further DNA gets replicated and divided. Next, central dogma is given, which says that from DNA itself getting replicated, it undergoes the process of transcription and then translation to make proteins which carry out our life. These three processes are the foundation of all the fundamental processes that we study in biochemistry. Next, we study the structure of DNA. So, DNA is made up of two words, deoxy and ribonucleic part, where deoxy is the sugar part, while nucleic acid is the macromolecule which stores the genetic material. It is attached to a phosphate, and the monomers are called nucleotides with the phosphate attached to it. Next, replication is the process of synthesis of daughter DNA from parental DNA by the enzyme DNA polymerase. Here is the reaction where monomers of mononucleotides are attached together to make a lengthened DNA. These are the nitrogen bases which makes the ladder of DNA. These four are very important from exam perspective because their numbering is often asked. Structure of nucleotide which is attached to the phosphate. Basically, a nucleoside when attached to a phosphate group makes the nucleotide, and this bond is a very strong bond where this is attached to a phosphate and it is called as phosphodiester bond, while the bases A and T are attached together by a very weak bond which is called as hydrogen bond, and C and G makes the triple bond out of it, while A and T are double bonded and they are basically very weak bonds. So C and G they have a triple bond while A and T are double bonded together. So phosphodiester and hydrogen bonds, many numericals are asked from this diagram and this structure. So you need to remember the structure of DNA. A and T together and C and G always together. Next, we will see types of DNA replication. Initially, while studying DNA as a molecule, it was studied that it gets replicated by these three processes in conservative, semi-conservative, and dispersive. In cesium chloride, density based gradient was made after replication, cesium chloride, and based of that N14 and N15, two types of isotopes of nitrogen were used to tag it, and in semi-conservative, it was found that a hybrid DNA was formed, while in conservative, both DNA were put together separately. General characteristics of DNA was stated, which said that it is semi-conservative and the replication is bidirectional and also not continuous. Semi-continuous and it occurs with very high fidelity, means very high proofreading. The process was proved by Meselson and Stahl in an experiment in 1958, who proved that DNA replication is semi-conservative. How? They took E. coli cultures in cesium chloride and they allowed their DNA to replicate for 20 minutes. They found in N15 they were getting pure DNA, but after 20 minutes next they getting a hybrid DNA of N14 and N15 when they transferred the medium to N14, and which was the light nitrogen, and then when they extracted the DNA it was hybrid and light DNA both after centrifugation. This clearly showed that after each generation, the parental strand was getting 50% distributed in their next generation. So, this was the ratio after 60 minutes. So, this clearly stated that DNA is semi conservative in replication, the old strand giving rise to new strands. and each strand is a template for the new strand, a complementary strand. Now, when replication takes place, it leads to unwinding of double-stranded DNA. This double-stranded DNA makes a replication fork when it gets unwinded and makes a particular point at origin site where the process of replication starts. Parental double-stranded DNA and two newly formed double-stranded DNA form a Y-shaped structure, and it is called a replication fork. So it is not a unidirectional process, it's a bidirectional process, and you can see the shape of fork on above side where origin takes place and two Y-shaped structures of fork are made. This is the structure which is gets made when a double-stranded DNA is unwinded. This fork is made and origin is also shown in the middle. And we should always remember the polarity of DNA strands here. 5 to 3 direction the fork moves. So in 5 to 3 is the template strand in which replication takes place. Next, we will discuss that why the process is 5 to 3 and why in 3 to 5 small these fragments are made. In prokaryotic and eukaryotic, different kind of replication takes place. In eukaryotic, because the DNA is linear and very long, the replication are multiple and origin of replication are also multiple. While in prokaryotic, because their DNA is very small, genetic material is in circular DNA state. So one replication and one origin of replication is there, and replication forks are made like that. First, we will cover prokaryotic replication in this video. So it is also called as a theta replication because the structure is like that, theta state. Here you can see, and the enzymes and proteins required in this replication are these. These are very important, and we will cover how they help in the DNA replication. DNA polymerase is the primary, and its role is to match nucleotides and polymerize adjacent nucleotides. In this diagram, you can see helicase. It is the enzyme which unwinds the DNA and melts it. It opens up the DNA. It cuts the DNA. Primase, which provide an RNA primer to start polymerization. It is the enzyme which provides the primer, small primers. Next, single strand binding proteins, SSB proteins. When the DNA is unwinded, it can't stay in single stranded form. Then they bind to the DNA and stabilize it so that it does not bind to any other complementary strand. Next is gyrase. Gyrase is a topoisomerase which relieves torsional strand. DNA, we know the structure. It is like a braided structure, so it open up the structure and it releases the torsional strain. Ligase is the one which join adjacent DNA strands. Here you can see how topoisomerase works. It unwinds. Next, we will study the DNA polymerases in prokaryotes. So, DNA polymerases in prokaryotes are of three types. We will study polymerase 1. It was found by Arthur Kornberg and it possesses polymerase activity and exonuclease activity like rest of the polymerases. Endonuclease means which cuts from within, while exonuclease cuts at the ends. Activities of DNA polymerase is that 5 to 3 direction it polymerizes the strand. In this diagram, you can see how pol 1 works. And in 3 to 5 exonuclease activity, it removes the unmatched DNA. You can see on the lagging strand, that is 3 to 5, it is removing the mismatched. 5 to 3, we know it is removing the mismatch, and 5 to 3 exonuclease activity is also there, which removes RNA primers. From 5 to 3, it has polymerase activity and also exonuclease activity. Means 5 to 3, it is making addition of nucleotide also, and it is removing mismatched nucleotides also. Now the whole DNA pol 1 structure is divided into two fragments, out of one which small fragment possess 5 to 3 exonuclease activity, while the large fragment, also called as clino fragment, is very important. It possesses 3 to 5 exonuclease activity. So smaller one has this. In this diagram you can see it has nucleus activity from 5 to 3, while clino fragment has 3 to 5 exonuclease activity, but it has polymerase activity also. This question is very frequently asked in exams, and you should remember this. Clino fragment questions are asked here, that the large fragment, what activity and what's its role. So DNA pol 1 having small and large clino fragment. 5 to 3, it has exonuclease activity, while the large fragment, clino fragment, has 5 to 3 polymerase activity. So you can remember that the large has 5 number, so large polymerase activity, rest has exonuclease activity. Next is DLA pol second. It is temporally functional only when pol 1 and pol third are not functional, and it participates and DNA repair only. Next is pol third. So pol third is a heterodimer enzyme which is composed of 10 subunits. In this diagram you can see it's the asymmetric dimer, means it's a heterodimer and it has 10 subunits and it has the highest polymerization activity also. All units are different and polymerization activity is 10 to the power 5 nucleotide per minute is its polymerization capacity, and it is responsible for elongation process which we'll cover next. So the pol third structure has alpha subunit in this diagram you can see, which has 5 to 3 polymerizing activity. Epsilon subunit has 3 to 5 exonuclease proofreading, and theta subunit is maintaining heterodimer structure. Basically it is for stabilizing it. So if you see the structure, it's a hand-like structure, it's a clamp-like structure which holds the strand. And here you can see the leading strand is shown where DNA is unwound by DNA B, which is the helicase of prokaryotes. Next we will see helicase B is the DNA B is the helicase here. And it is the core DNA pol third structure and how it binds on the leading strand. Now, how nucleotides are added on the strand. DNA polymerase always add nucleotides at the three end of the growing DNA strand. It is the structure of sugar you can see, and O is the free OH hydroxyl group where coming nucleotides are added. The free OH group forms a phosphodiester bond with the incoming nucleotide, which is a very strong bond. We already know. So here you can see on the leading strand on the 3 group how it is added. Now RNA primers which are added while polymerization. So here primase is the DNA G which synthesize short RNA primers using free NTPs and single stranded DNA template. Here you can see the DNA template, and here primase is there which is DNA G, and on it RNA primer is added on the 3 end for extension by, and DNA pol third here polymerize it and catalyze the reaction, and at initiation new DNA is formed like this. So primosome complex is formed by primase DNA B DNA C at initiation, which we will cover. Next is helicase. Helicase here in prokaryotes is DNA B, which opens up the double stranded DNA. In this structure you can see the circular helicase. DNA B is there. Single stranded proteins we already know it stabilized single stranded DNA. DNA gyrase, which is a topoisomerase, is in both strands to relieve supercoil. It reforms phosphodiester bond and use ATP while doing this reaction. So it is basically an energy consuming process. Gyrase is present here and it uses ATP, and negative supercoiling is there. So gyrase also use ATP. DNA ligase join the adjacent single stranded DNA and seals next. Next, we will cover the stages of replication. It is in 3 forms: initiation, elongation, and termination. Initiation occurs at origin of replication, and here double-stranded DNA is separated and primer are synthesized. Elongation and termination also we will study next. Gyrase, you can see initiation process, how helicases are loaded, DNA B and primer are synthesized. And lastly, DNA termination at the end of DNA. Prokaryotic origin takes place at ORI site, ORI C, and origin is at 248 base pair long and it is 80 rich. If we see on the leading strand, then 248 base pair long origin is there, and it is composed of 13 base pair sequence and 9 base pair sequence. So Ori site comprise of 13 and 9 mer sequences. They both are 80 rich. Here you can see the base pairs. It is 80 rich and it is 248 base pair long. Now initiation process involves pre-primosome formation here. DNA A, the protein recognize the Ori C. Now DNA B, which is the helicase, it and DNA C join this DNA A complex and open the 80 rich region, means This helicase opens the AT rich region, which is the origin site region, to initiate the process of replication and separate these strands. DNA A is gradually replaced now, and SSB proteins bind to stabilize the single stranded DNA. So basically, helicase and primase together form the pre-primosome complex. Now, primase binds and forms primosome complex. Primase synthesize RNA primer on single stranded DNA. Primase is responsible to synthesize RNA primers needed on single stranded DNA replication. Using NTPs in 5 to 3 direction at expense of ATP means gyrase and primase both use ATP to function, and RNA primers provide free 3OH for DNA elongation. Now primase has joined the primer. Now further nucleotides are added by DNA pole third. Core enzymes of pole third include alpha, epsilon, and theta. They catalyze leading and lagging strands both. Okazaki fragments are made in the 5' and 1000 to 2000 nucleotides are there in prokaryotes. It is the lagging strand, and directionality is 5 to 3. We know of leading strand. Last process is of termination, where both replication forks meet. Here we know circular DNA is there in prokaryotes. Ori is the origin sites, and here a replication bubble is formed because of two replication forks. It is bidirectional, we know, and both forks now will be meeting at the same point. So they meet at the ter site. Here in the opposite side, you can see ter sites are present after the end of replication, after originating from here and coming at the last end at the ter sites. They are meeting at 32, and fragments of DNA here are ligated by DNA pol 1 and ligase. They both help to join these fragments, which are at the end coming, and ter binding proteins are present, which indicate the termination of the whole process. So this was all about replication in prokaryotes, and it is very important, more than the eukaryotic replication, which we will cover in the next video. So thank you so much for watching, and like, share, and subscribe.